body. What is it?
Fleming of St Mary's Hospital, London, who discovered that it produces the drug known as
The
penicillin.
question was, which drug would go with which cell?
It's a collaboration that's unexpected because it's between humans and the tiniest organisms.
The howl of a dire wolf.
Dorset lamb may represent a major landmark.
I did not start off planning to design proteins.
Scientific breakthrough will probably be something that we hard to imagine.
Test at birth could map out an individual's entire future risk of disease.
Well, Michael, welcome to the show. You're obviously the co-founder of Oden Labs, and I think that's a good place to start. Tell us a little bit about like why you started this and what was your journey to doing it.
Yeah, so Oden Labs is an attempt to build the first ever automated animal research lab. And as you guys probably know, there are a bunch of automated biolabs right now, but nothing like this ever for animal studies. And the way that this came about was actually as a result of my PhD, where I was building gene therapies for longevity. And as part of that, I would have to test hundreds of animals for the effect of these therapies. And that was such a laborious process. I spent literally almost a full year doing nothing but just manually measuring mice all day, every day to get like a very basic data set. And so the summary is I discovered this bottleneck on my own skin, looked around in the world, discovered that everyone else is doing the same thing. And this is despite the fact that animal studies are literally the backbone of drug development. Like up until now, everything that goes into humans had to go to animals, but we are measuring them one by hand. There's no automation, there's no digitization, and the data quality and the data amounts are just very, very, very poor compared to the rest of the biology, which has been digitized. So yeah, I realized that if this bottleneck wasn't solved, we as a civilization couldn't make substantial progress in developing better longevity therapies, but like therapies overall. So yeah.
Got it. And just to touch on this point a little bit more, like just animal studies in general, very, very complex, very, very difficult, lots of manual labor. A human needs to feed them, a human needs to take care of them, remove their poop, like all of these things. And then also be tracking what the impact of the intervention is on the animal. And I think right now there's like an email system at most labs where you get like emailed whenever your mice dies or if there's like a major update with your mouse. And that's like someone physically going there, looking at your mouse and then writing you an email. So talk to me about like, is that the only problem? Like what's like the broader problem? What are the real sort of annoyances that researchers currently have to deal with when doing these animal studies? And then, yeah, like how do you possibly solve this through automation?
Yeah, so I think there's three major problems in the animal research space. The first one is just the pure cost of the animals itself. So for example, right now, if you want to buy an aged mouse that's a year and a half old, it's somewhat equivalent to like a 60-year-old person. It costs you about $700 in the US. Now, for a regular longevity study, you usually need about 100 of these. So right off the bat, it's $70K up front before you can do any experiments at all. Right? And guess what? They're sold out. You can't even access them. And this cost comes almost purely from the fact that as of today, every single day, a human needs to go into the mouse house, physically look at the mice, physically change their food, physically change the water. Sometimes it's not even an automated watering system. They have water bottles, literally, that a technician has to go and like change. And it's just insanely laborious and therefore expensive, right? So that's the first thing, just the cost of the animal because of the cost of the human labor. Second thing is the ability to gather data and the quality of that data, right? So as I mentioned, when you put in a therapy, you ask like, does it make the animal better? Does it make it worse? And if so, in what areas? And to know that, you have to really measure across the body, like what's the cognition, what's the strength, how active are these animals, like how social are they, do they take care of themselves, do they eat, do they drink, do they sleep well enough and so on, right? Pretty much everything that you want to measure. Right? And as of today, every single thing that you do, a human needs to go pick the animal out of its cage, put it on a test setup, train the animal, usually a couple of days in advance, then do the test multiple times because there's a lot of variation, and then repeat this for all the animals. And it's just, you can't scale this. It's stressful on the animals, and the data quality that you get is highly dependent on who's actually doing the measurements. So there's these great papers that show that I think you're better off being a male than a female as a human when doing these things, because for whatever reason mice seem to be like more stressed out with females, maybe because women sometimes wear like perfume or whatever. Like there's like very clear human hand effect things, and I saw that as well. We outsourced one study. Biggest effect size was a clustering by the technician that was doing the measurements. Right.
Fascinating.
So there's that, and as a result, like you can't scale. Again, it's expensive, and studies are not reproducible in the sense that you don't even know if what that person gathered is going to be reproducible by another person. And then there's the third big aspect, which is the humanization of animals. And that comes down to the quality of the model, right? So, for example, if you have a disease, perfect case is Alzheimer's, not a single Alzheimer's drug out there in the market after 35, 40 years of research that actually works, right? But there's a bunch of different models for Alzheimer's that mouse models for Alzheimer's that maybe tend to recapitulate the human Alzheimer's disease. Well, in this case, it's not a good model at all, because these animals are they're like over-engineered to produce something that kind of looks like an Alzheimer, right? And that's even in a case where we have, these models exist. There's many other cases where we just don't have a model for a specific disease that replicates this. And largely this is because it's very difficult to engineer animals. Like right now, if you build a new cohort of animals from scratch, that takes you about 1.5 years just to get to your first homozygous animal that has both copies of the genome, has your intended genetic modification, right? And so people tend to default back to these existing models that someone else has somewhere created that they can just like buy off the shelf and then try something. But usually they're not very good, or not usually, but there are areas when they're not very good or when they're not very appropriate. And I'm not saying that the mouse is going to be that is always going to be possible to create an accurate mouse model of a human disease. I think there are areas like Alzheimer's in which the case is very difficult. But there's two things that you can do to improve the odds a lot. First thing is you can put in human genes or like humanized versions of the genes. And I think a very good example here is Regeneron, the massive pharmaceutical company. They spent literally 25 years coming up with a new system to humanize the mouse, B cell and antibody production, right? They had no drugs for 25 years. Then they figured it out by putting something like 2 megabases of DNA into this mouse. I think it's called like Velocimouse or something. And then since then, every single major drug from the data that I saw has been a hit, like 8 out of 8.
Wow.
And that's insane. Like, it just doesn't happen. And it's actually very interesting because the author of Room's law, if you've heard of it.
In terms of the cost of clinical trials.
Yes. Yeah. So Moore's law is like the cost of compute drops like by twofold every 2 years or something like that.
Something like that.
In pharmaceuticals, it's the opposite. Like the cost or the probability of getting a drug per unit money like increases twofold every year, like since like 1950s or 1960s or something like that, right?
Yeah.
So it's like it's getting worse.
It's getting much more expensive
Yes.
to get drugs through clinical trials as time passes.
Exactly. Right? And the question is why? And so the author that discovered this from data also did a meta-analysis where he said, compared to the quality of the model or the predictiveness of the model versus how many drugs you screen or test, if you improve the predictiveness of the model by 1%, that has the same effect as screening 16 times more drugs.
Okay.
Right?
Yeah.
Somehow came out of his math. So like model predictiveness is super important. Yeah. And a way to do that better is by putting in human genes, which is what the whole body gene delivery system helps to do. And the other way is by putting in human cells in a smart way. Right?
Got it. So let's take a step back here and touch on like a few important points. One is that you mentioned that a lot of the sort of disease models that we use, especially in mice, don't perfectly or even sometimes even slightly accurately recapitulate the etiology of disease in humans, right? Like the Alzheimer's mouse models, like somewhat simplistic representations of Alzheimer's, or they might capture like individual facets of the etiology. And there's like still a debate in among scientists around like what exactly is the upstream cause of Alzheimer's versus sort of these downstream presentations and aggregates and whatnot potentially. So that there's that. And then there's a bunch of diseases for which we just pretend like we have mouse models that recapitulate, but we really don't. I mean, we have sort of you mentioned aged mice, but there's also like these aging mouse models that have been engineered to age more quickly, sort of accumulate mutations more quickly. But then those are also very flawed, and they need to improve in order for us to be able to do aging experiments or test aging interventions. Can you talk a little bit about all of these kind of like models and what's holding them back, like where we need to make progress on that front?
So I think first thing is, I think no model is flawed. The humans running the models and trying to interpolate something wrong out of them are flawed, right? It's us, it's not on the models. Every model is doing something. Now the question is it doing what you think it's doing? And is this the right thing for you to ask? So for example, like these accelerated aging models, I think they can be super useful. For example, I don't know, it's like, progeria mice, like the actual progeria, the what's it called, Hutchinson-Gilford progeria mice, they have a mutation in the lamin protein that messes up the nuclear membrane, and that causes like downstream effects in many different ways. Well, that is one aspect, like it's one de facto aspect of aging, right? It's been maxed out in this model. If you want to study that aspect of aging, that's actually a very appropriate model, right? Like, for example, nuclear integrity or like nuclear membrane integrity. If you want to use that to study, let's say epigenetic reprogramming, yeah, maybe it'll allow you to do some things better, but maybe not, and maybe it'll lead you astray, right? So I think that's the first thing, which is there's no bad model and there's no perfect model, right? It's about the human that's doing this. But I think the second thing is, and this is the big picture takeaway here, most models have been limited to mice as a model organism because, to our first approximation, they're really the only ones that we can engineer right now. Like, you can do some things in monkeys, you can do some things in other species, but it takes so long and it's so expensive that practically it's almost never done. And that's because the generation time of a monkey is like multiple years versus for a mouse it's like 3 months. And also the genetic engineering tools, like the germline CRISPR-Cas and like whatever, just aren't there, haven't been built. And if we had a system that would just allow you to take an existing animal of any kind, put in DNA broadly across its body, and quote unquote turn it into transgenic, now you could do many different kinds of animals, and you could choose your models in a much more sort of sophisticated way, right? So yeah, I think most models, they are not wrong. They're just, we have a limited number of options, and the fact that we're limited to mice itself is also a major issue, and it still takes a lot of time, right? So there's just no way around. Yeah.
Okay. And the other thing you mentioned was around improving the correlation between the assay or the model that you're using and the clinical endpoint that you want to achieve, and that if you can even shift this by 1%, then the amount of sort of lead candidates that you are going to be trialing in clinical trials, for example, you're going to get a much better success rate out of that. Could you talk a little bit more about that?
So this is, I should go back to the paper that this guy published, and I'm forgetting his name, sorry. But this was a basic mathematical calculation that he did. It was like it wasn't, I think, observational. It was more just like statistical and mathematical. But he said was, given these assumptions, if you have a model that recapitulates the actual disease 1% better, or maybe like 1% more often or something like this, versus the more things you screen, then you should always try to like improve the model. Right? Yeah. I can find out the paper and send it to you later.
And I think that just makes intuitive sense. Like that we're currently using, even like let's say in like small molecule drug development, if the assay that you're using is not like a good proxy for the clinical endpoint that you want to achieve in a clinical trial, then you might screen 100,000 drugs or 1 million drugs, but
you're just doing it for the wrong thing.
You're screening for the wrong thing.
Exactly.
And so there's going to be a disconnect, and you're going to result in failures in sort of phase 3 clinical trials, for example, much more often. And that's like we also have talked about the rise in clinical trial costs. And one of them is sort of like animal studies. But do you have a sense of like what is causing this kind of really negative trend?
So actually, animal studies makes up a very small amount of that cost overall. Like I think 70 or to 80% of the cost comes from phase 3 trials, which is where you really amp up the numbers of human test subjects, and you also have to amp up then the drug production and everything. So, we're talking about like low 1 or few percentages in animal studies. But then why is that so important? Well, it's because once you have a drug in humans, you can't change that, right? Like that's it. You just have to keep on going or it hits the wall. So the real development happens actually in the screening and then the animal testing level. But actually, to add to that point, FDA does not require animal efficacy data. It requires animal safety data from different models. And as a result, many times you don't even have an animal efficacy being tested before it goes into humans. Right? And maybe you can argue, yes, given the current state of affairs, with the quality and the cost of models and the time that it takes to run them, maybe it makes sense. But empirically, we see that there's a 90% failure rate, actually 90.6% failure rate. And therefore, it clearly isn't all that good of an approach. So I think
what isn't all that good of an approach?
Not doing efficacy testing in animals before going into humans.
So you should do it.
You should do it. Of course, you should do it. I mean, like, it's easy to make something safe. It's just, you give cyanide at low enough dose, it's safe, right? But doesn't mean that it's gonna, treat anything. But I think, again, I'm not an expert on why clinical trials are so expensive and why they have been getting more expensive. My guess would be it's because of the cost of all the failures and the cost of regulation. And maybe also the fact that many of the low-hanging fruit that do not require things like biologics, gene therapies, cell therapies, have already been kind of hit by small molecules. And so every next phase requires something more difficult.
Got it. So let's talk about what is in your control with Oden Labs. So smart cages, or at least these like smart lids that you put on established cages that are already being used in animal facilities. How did you arrive at this, and what are these cages? What are they able to do?
Yeah. So first, the question was, okay, mice are limited, but they're still good models for basic biology and for certain things. Like even Alzheimer's, like you can argue that they actually make the amyloid protein really well. It's just that the amyloid hypothesis probably isn't the right one, right? But the question was, we don't even have the basics of, in this limited mouse model, is this reproducible? And does it even work there? So how can we make that happen? And when I was thinking about this in my PhD, I realized that the only way to do that and to scale is to move away from the need to move the animal out of its cage and put it on some new test setup, for example, to measure its endurance running. Right? So like a good example is you take a mouse out of the cage, you put it on a treadmill, runs, and you put it back, and then you do it again multiple times. And just, it is fundamentally not scalable because you can't solve the logistics. So what do you want to do? You want to measure everything that you want to be able to measure directly where the animals live, like in the home cage. And that's pretty tough. And for various reasons, it's, there's things that you need to hit. But that was the goal. Like, how can we measure all aspects of health that are actually interesting directly inside a home cage?
Okay. And then how did you do this? Like, what's the technology in the lid? What are you tracking? And then where does all of this data feed into?
Yes. So right now, what we have launched is a kind of broad use, quote unquote, smart lid. And it's an adapter that fits above an existing cage in an existing rack, collects video and audio data, then runs this data to a bunch of neural networks and basically a computer vision pipeline analysis, tracks every one of the multiple mice individually, and then extracts 23 different metrics of health and behavior. That's what it does. Right now, it's based on vision, so vision and audio. We can also put in things with where the animals interact with them, and then we can get things like weight, like strength, like cognition, and so on. But that's what it does.
Got it. And then sort of besides this visual inspection, these vision models that are running constantly, what other what's the pipeline for the future? What else can you track besides weight? Are you going to add like robot arms inside the cage that like move the animals around? Like, what's the plan?
Yeah, so inside a cage, we're going to do very little. There, it's mostly about how can you capture as much data as possible with minimal changes to the infrastructure or to the actual cage itself, right? And so there's a bunch of things there. For example, people that are interested in cancer, they want to quantify the tumor volume. For that, you need a 3D camera. It's a bit of a different hardware. For people that are doing neuroscience, they want to do cognitive testing. For that, you need to either have a touchscreen based assay setup or something else that you put in the cage. So there's these pockets of things that currently specific subfields require. Now, in its extreme, what I want to do is pull like all the 80-20 of these together into one system that can fit into the existing infrastructure and kind of establish this phenomics field, which is how do we gather the omics of health and behavior in a constantly in a structured 360 view way. Now, when it comes to the handling of animals, or like the robotics that you mentioned, that is a bit of a different problem because there what we're trying to do is solve the animal care cost and scaling issue. And basically, somehow you need to do away with the human. If you have a smart lid in the cage, you can see exactly how what's the cage status. Does it require a change of food, water, or bedding? Are there any sick animals? And once you have that, now you can link it to a robot and say, look, robot, go and do this one thing that needs to get done, or even do a cage exchange. And the difficulty there, beyond not having the eyes in the cage, was, well, you don't want to have the robot grab a mouse. Like, that's a very stupid idea for many reasons. So we had to figure out a way how to do all these day-to-day care items without the robot actually ever physically interfacing with the mouse. And there's a way to do it. So that's how it's going to be.
Okay, got it. So you're imagining, and phenomics comes from like phenotype omics, right?
Yes.
So you want to, like, you're imagining long-term, fully automated, large animal studies being done where you have some mice that are controlled, some mice that have been given the intervention, and you're tracking, are these mice that have been given the intervention more healthy? Are they living longer? Or do they have more energy? Is their sort of cancer tumor size decreasing or not? All of this in like a more automated way, with there's still like humans involved in taking care of like the feed and changing the water, but sort of the monitoring at the very least is taken care of, and the data collection is more objective, more reproducible, right?
And more scalable. Sure.
Got it. Okay. And then like very, very long term, do you imagine these just like large warehouses of like smart cages with like humanoid robots walking around and changing the food? Like where does the robotics come in? Right?
Yeah. So theoretically, we can do robots today, but it's you want to solve one very hard problem at a time.
Yeah.
So that's why even though we've designed it, we haven't launched them yet. But so the robotics, maybe you can do it with a humanoid, and if we wanted to service the existing facilities, we would probably do it this way. It's better to do it not with a humanoid way, but it's like actual purpose designed robotics that do the movement of the animals in the care. And so the way that I foresee this going is, right now we have this smart lid that fits into existing systems. we are directly selling this to people. They can access this, they can run their own studies. The next phase of that is doing an in-house cloud lab where people that don't have a lab can just outsource their studies, and then they get the smart cage data. But we also have our own robotics at the back end to like handle the volume and the logistics. Plus, if you have the robotics and you let them run for a couple of years or more, now you can start bringing down the cost of aged animals, and theoretically by a lot, like from 700 maybe down to like 100, maybe even like less so. And that would very likely allow substantially more experiments to be run by the people in the field right now, which is when you ask them like what would you want to run, they tell you all these things, but they can't do it because it's too expensive. Yeah.
Got it. And the reason that you want to have specifically aged animals is for more of these age reversal type interventions like reprogramming and whatnot.
Well, so there's the age reversal, but there's also the speed of anything. It doesn't even have to be reversal, it could also be protection. And even just like basic biology studies. And I think this is a good point to cover, like, given that animal models are imperfect, why do them at all? Why not just do like organoids or human cell lines or whatever? Well, the reason is very simple. We don't know enough biology. And things like longevity or like aging and neuroscience, they require a full physiological system of which we do not understand enough to put it into any kind of model right now. So there's like absolutely fundamentally required, definitely for basic science. And for example, if you want to know, well, what happens if I don't know, have this new reprogramming factor that I've tested in a cell line, but I don't really know what the effect of this is. Well, you have to run a mouse study or some sort of an animal study and see, are they better? Are they worse? Do they live longer? Right? Very basics. Now, you could start that from day one when you make the mouse or when you make the animal, and then wait for them to age for 2.5 years. And then you have to wait for 2.5 years. Or you could start with an older cohort of animals and go straight into a point where they are already aged and maybe do the study in 6 months. You might have a lower effect size because you started when they were already sick, but you could get your answer much faster, particularly if you add more numbers of animals. So
in the sense that aging accelerates the older you are, and therefore an aged mouse will more quickly show you the results of any intervention.
Exactly. Yes.
Okay.
Yeah.
Got it. Very interesting. And then you also mentioned something to me, like even more longer term than this, in like the sense of like the long-term moat around the product will be the data that is collected and the models that can be trained on that data. Tell us about that.
Yeah. So I think none of us wants to do studies with animals, right? Or definitely not with humans for that matter. Like ideally and at its limit, biology will be fully understood, modeled, and everything will happen in silico, right? Question is, what do we need to do to get there? What do we need to measure and how much data do we need to have? And we now know from different pockets of biology that it's possible. For example, AlphaFold did this with protein folding, one very straight up problem. Then various SCGPT models have done this with predicting like what's the effect of a mutation or like an intervention on gene expression, right? And this happened because there was very large amounts of highly structured and high quality data sets out there in the public upon which different AI models could be trained. Right? So for us to accurately model a full physiological system, like an animal or like a human, we need to have physiological data that is similarly, very large scale and very highly structured and just like high quality, right? And the only way to do that is to have a system where we have some sort of an automated way of measuring the health of animals and doing quote unquote phenomics on them, map out, what happens to every single aspect of health, and then just have the community repeat it over and over and over again. Like, I don't think any single facility or organization is going to be large enough to do that, right? The only way to get there is to have a game theory in which for everyone involved, it is actually best for them to use this automated system and put the data into one place, right? So that's what you got to build, and that's what we're trying to build. So if we get this right, it's better than a human. gathers high quality data that's structured and the same, and ends up in one place. Now we have all the bits of the game theory there to have this large data set to be built upon which ultimately we can model the mouse. Right? And of course, if we can model the mouse, then it's a small step to modeling the human. Because biology is like 95% shared, or however you quantify this. But it's getting to that first step, which is the most difficult one. And then you can use transfer learning.
Got it. Yeah. Okay. So it's somewhat like trying to develop like the protein data bank of animal studies. And if you took this out to its limit, like what would that mean? That you just like have a molecule, and then the model that has sort of been trained on every intervention in animals and sort of tracked their phenomics over time will just be able to tell you if this is going to be successful?
Yeah. Okay. I mean, I think that's where they all lead to. And I think the animal data set would be one major part, and maybe the last part of this general biology foundation model, where now we know that if you have drug X, you can do protein modeling and see where does that drug X hit. So you can do like in silico screening, like that's one way. And now if you know that, okay, we can hit this protein X using the SCGPT type models, we can say this is going to be the effect of gene expression across these cell lines. And now if you have that data coupled with animal data that says once this drug X that hit protein Y got into animals, we have health changes in X, Y, and Z behaviors and aspects of health, then you can say, oh, you know what, with this specific change in expression pattern, these are the health outcomes. And then you just keep on doing it over and over and over again. And hopefully by the time you get to like 10,000, 100,000, a million iterations, you just have enough pattern recognition to say what's going to happen next.
Got it. Yeah. So it's not that you think that just the phenomics data itself is going to be sufficient to do this. Yeah. There's like this like data aggregation across genomics, across single cell RNA-seq, across these other models such as like SCGPT that basically predicts like the effect of methylation patterns on expression, right? Yeah. Okay, got it. Yeah, I mean, a big. We'll see, right? Like if this pans out, hopefully that would be great. I want to take us somewhere else, which is obviously in your PhD. You actually you're a genetic engineer. You're focusing on delivery. Like what got you into that space? Tell us about like a little bit about your research and your PhD.
Yeah, so the PhD is I suppose a natural continuation of what I started doing when I was 16, really. And that was to build the tools to engineer humans to live longer. Right? And particularly by the time of my PhD, I got to the point where I knew the synthetic biology, the genetic engineering. Now the question was, how do you get this into the animal and what do you do once it's in the animal? And so therefore I focused on two problems. First was the delivery, particularly how can we hit the whole body? How can we have a whole body gene delivery system? And secondly, once we have that, like what are the genes that we might want to hit that would increase healthy lifespan? Yeah.
Got it. So on your LinkedIn, you write that you've been genetically engineering longevity since 16 years old. What does that mean? Like, what did you do at 16? Yeah. Why have you been in the lab for so long? What happened to your childhood? Right?
It's a lost childhood. Man, it's just I was a kid and then I became an adult. There's no teenage years. So what happened was, I was looking for a meaning for my life, and I still remember I had discovered the theory of evolution, like when I was like 13 or 14, and theory of evolution says life does not have an inherent meaning. Like we're just here because our forefathers survived, and so I had this depression. It's like, look, what am I like? Why am I here? And at first, I was very interested in physics. This is what I thought, biggest impact of your life, that's what you can do in physics, right? But realized very quickly that actually it takes you a very long time to do anything significant in physics, and you kind of need to get lucky, right? So effectively, when I was 16, I was trying to figure out what is the best thing that I can do with my life, with the one life that I have, and make sure that even if I wasn't successful at this, it would be a life well lived, and I would have, been going in the right direction. And I remember when I was, so a year before I think my sophomore year, I suppose it's what you call in high school, like 9th grade, 10th grade, I discovered two things at the same time, almost back to back. The first one was the vacanti mice. So the vacanti mouse is a mouse that had a human ear grown on its back.
Okay. Yeah.
And like most people look at this and it's like, oh, this is disgusting. And yeah, it's kind of disgusting. But what it showed me for the first time was, as a civilization, we are there with biology that we can actually engineer it, like fully. And secondly, I came across Aubrey de Geer's book, Ending Aging. And there he argues that aging is actually just an engineering issue. And I still remember he said something about gene delivery on the order of gene delivery would be great, but it's like way too far into the future for it to be like effective or something like this. I was like, I'll show you. So in any case, I discovered what I thought was the most impactful technology and the biggest problem of mankind, which is aging, age-related diseases, and they kind of fit together like one by one, right? And so at that point in my year, then the question was like, but what do you actually need to do this in humans in a way that's actually effective? And I mapped out the skill set that I know that I needed to have. I got into the lab that year on my first genetic engineering project. I was super lucky to have this biology teacher who had a lab in Tallinn Technical University. And so I just got into the lab and started working on genetic engineering E. coli. Really.
This is in Estonia.
This is in Estonia, yes. Yeah.
Okay.
And yeah, that's where I started.
Got it. So you read Aubrey de Grey's book. Aubrey's coming on the podcast at some point soon. And this kind of opened your eyes to the world of aging, and this can be really impactful. Like everyone is aging. A lot of diseases are age-related, and if we can sort of address the upstream causes of aging, therefore we can prevent or mitigate a large fraction of the diseases that cause the suffering of mankind. And then also, you saw this mouse with a human ear growing on its back and realized that, like, wow, we're, as a society, as a civilization, we're approaching this phase where biology has become engineerable, right? Like these genetic engineering tools, these delivery tools are getting really good. And Aubrey was saying delivery is the bottleneck, right? And then so you thought, well, these are the skill sets that I need to develop.
Yeah.
So what was the roadmap from there? What did you learn? What do you still have left to learn?
Yeah. So I think at the very basics, I knew that I needed to learn biology. Like up until that point, I was, dead focused on physics. So that's where I started. That I would need to learn basic genetic engineering, which is somewhat different from the rest of molecular biology, and then that you would have to learn synthetic biology, which is like genetic engineering on steroids. Like how do you design entire circuits to do something? That then I would need to learn to do this in the human cell, which is very different from the bacterial cell, where most of the synthetic biology had been done at that point. And then finally, I would need to learn to do this in the human itself, or in this case, in the mouse, and figure out how we can get this in the in us while we're already adults, and then how can we work with all the constraints that come out of this requirement, given that we can only put in like small bits and pieces of DNA, and that you have issues overcoming the immune system and so on, right? So that's the basics. Now, of course, if you want to develop a drug, what I haven't done so far is run clinical trials. That's one more thing. And arguably protein engineering, which you might want to have if you wanted to do your own like enzymes. But yeah, I think at this point I would probably hire these competencies in rather than taking another 7 years to do a PhD and another one.
There is a balance between like how many skill sets you need to develop by yourself versus like other people that can help you, right? Again, like the civilization as a whole, we figured these problems out. It doesn't have to be all you, right? But yeah, so you've touched on the importance of genetic engineering, which is like a slightly different toolkit to molecular biology. Like there's certain sort of niche specific things that you need to learn in that field. And then yes, it's synthetic biology in the sense of designing gene circuits that get to do what you want them to do, that sort of manage expression levels of any gene that you want to insert, and then delivery, right? So delivery is what you worked on in your PhD. Can you talk us a little bit about like what are all the challenges in delivery? What are all the tools that we currently use as a civilization to get genes into cells? And let's touch on that first before we go deeper.
Yeah, so there's, I suppose, three main challenges to delivery. First thing is just getting the physical molecule across the cell membrane and across the nuclear membrane. Forgetting about everything else, just the biophysics of it. That's one. Second thing is the adaptive immune system, which is your B cells, your T cells, your antibodies, everything that's in the blood. And then third thing is the innate immune system, which is things, molecular defenses inside the cell itself that recognize viruses and other pathogen-associated motifs and block that. So these three things you need to solve in delivery. What's the other question?
Yeah, I mean, what's the toolkit right now for delivery?
Yeah, there's a lot of different things that you can do. And many of them really only work in vitro, like when you have cells in a dish, or locally, when you inject them in vivo into an animal. But if you had to break this up, in summary, it would be viral vector-based methods, like AAV, the adeno-associated virus methods, like lentiviruses, like adenoviruses. That's one. Then you have LNPs, so lipid nanoparticles and their versions, like proteolipid particles, like so on and so forth. Then you have, I suppose you could call like RNA, like naked RNA or naked modified DNA delivered. That's somewhat chemically modified. Doesn't really work that well, but for certain things you can do that. And then
basically there's no vector at all. It's just the DNA straight into the cell.
Straight into, or in some cases
modified to be sort of shuttled across the membrane.
Yes, or even more often just the protein itself. So Cas versions that have been fused with like fusion peptides and then just like injected into the bloodstream and so on. So that's the third way. Then you have like liposome or just like chemical-based methods. These are lipofectamine, calcium phosphates, and things like this that just physically bind to DNA, remove its charge, or rather neutralize its charge, and allow it to cross the cell membrane. These are mostly used in vitro right now because they're pretty toxic. In vivo, you can kind of do it, but you're going to cause a lot of damage. Then there's electroporation-based methods, and that's you can do this both in vitro and in vivo. It's basically just zap the cell like hard. That creates a pore, and your DNA can passively diffuse into the cell. And then there's other things like lasers or gene guns or whatever, where again you make a physical pore into the cell, but they only kind of work in vitro. Yeah.
Got it. Yes. So out of all of this, what are the promising avenues for gene delivery in humans, and then what's sort of holding us back?
Yeah, so I would say definitely the most promising ones and objectively the most effective ones so far are viral-based methods. And out of that, AAV stands out by far because it is the only platform right now where you can get whole organ delivery in an adult human. And you can do that at a safety that is acceptable, right? Everything else, like lentiviruses, like adenoviruses are actually cancer therapies. They cause such a strong immune reaction that you can only inject them locally, and then that helps to kill the cancer. Lentiviruses, you can do them ex vivo, so you take the cell out of the body, usually like a blood cell, treat it with the lentivirus integrates, put the cell back in, and then it kind of does its thing.
And this is the mechanism for producing CAR T cells, for example.
Exactly, yeah. Although CAR T cells you can do in a bunch of different ways. So that's on the viral side, and then now the competition that's coming up is LNPs, so lipid nanoparticles, or more like they're the derivatives, like proteolipid particles. And there, in particular, the work from Oden Bio, and so it's not run by Matt Schultz, but he's one of the co-authors there, is, I think, the most promising that I've seen, where I think they modified the lipid bilayer with a certain protein, and they get somewhat, they get fairly broad distribution across the body. Now, I don't think, or at least I haven't seen evidence that this is as good as an AAV in terms of like vector to vector comparison. And in my work, I looked at like hard copies per cell. They didn't have that, so I just can't do the comparison right now. But it definitely seems very promising. And the advantage there is that you don't have to make a cell make the virus. If you can make the lipid nanoparticle with a mechanical system, it can be much cheaper. And theoretically, it can also cause lower immune reactions. But the disadvantage is, up until I see the evidence that like fully convinces me and is replicated by other labs, is just very, very, very difficult to come up with something that is as good as a virus that has had the entire planet to evolve for millions of years to get DNA into the cell in a way that doesn't like cause immune reaction or causes minimal immune reaction, right? So like the barrier to get there, considering all the limitations, is actually very high. And I think to a certain extent people underestimate that, but yeah, these are the two main ones. Yeah.
Got it. And I'd say like the core disadvantage of LNPs is obviously they worked for the COVID vaccines because it's just like intramuscular delivery and it spreads in like the local area. But if you want this to be delivered systemically, they're lipids, and so they naturally aggregate in the liver. And so you have this huge expression in the liver, not much in other organs. We've produced some other LNP formulations where we change like sort of the composition or identity of the fourth lipid or add a fifth lipid, and this allows sort of tropism to different tissues, but like not substantially. Like it's still primarily in the liver, and then you might get like a little bit of lung or like a little bit of kidney, for example.
But so that's why Ocean Bio's work was really cool, because they added proteins on the lipid surface, and to an extent enough where liver is still the highest, but it's like doesn't absolutely dominate anymore. You have other tissues as well that do get seemingly substantial amounts of DNA. And I think like with AAVs, again, many natural AAVs home into the liver because just the biophysics of the blood flow ends up pushing everything through there, and it's basically like a sponge, right? But now there are both natural and engineered serotypes that do not do that anymore, because AAV capsid is protein, and you can very quickly and very precisely engineer the protein, and enlarged in many different kinds of versions, right? Whereas if it's a lipid, you are limited to chemistry, and you're limited to basically to hundreds, maybe like thousands of different variations. With a protein, you can make a library of these, and you can have 10 billion of them in one tube, and you can test them. So that's the difference. And I think, again, they can probably meet together at some point, but yeah.
Got it. And let's touch on LNPs a little bit more before we transition to your work.
Sure.
Which is, we can change the protein corona around LNPs in order to get them to sort of detarget from the liver and to be retargeted back to specific tissues. How do we do that? Like, what are proteolipids? What do we physically do to the LNP to do this? And like talk a bit more about like the work that Oden has been doing.
Yeah. So I mean, they should be the ones here talking about this. So I'm just, I read their papers. I can give you the second-hand account. From what I understood, they tried a bunch of different fusion peptides that are small transmembrane proteins that kind of cause the cell to fuse with the membrane, or the LNP to fuse with the cell membrane. And apparently that alone, and I think they probably screened, maybe I think in the low hundreds or so, different versions of these. That alone was good enough for them to find one that worked well enough so that it didn't only end up in the liver, but it was like fairly broadly distributed, right? That's, I think, what they did. Now, I'm not 100% sure because I don't have the latest on this, and this is their paper. But I think once you have that done stably, you can build on top of this, and you can now have, I don't know, something like a nanobody unit that like faces out, where you can rationally engineer that or like semi-rationally engineer that against a specific transmembrane protein in a specific tissue that you can say, okay, if I don't know, my brain cells are expressing, I don't know, Ly6A, I'm going to target Ly6A, therefore the vector should end up in these cells more often than anywhere else. Like that's one way. Practically, and that's also what you can do in AAV, in that sense it's not different. But practically, what people have done so far, and I suppose could be better for worse, history will tell, is they have just mutated and screened. And like in AAV, you start out with a library where you make certain mutations along the capsid structure. And that just causes a conformation change of some sort. You don't really know what it does, but then you test and you inject the entire library, and you either in vitro or in vivo, and usually in vivo is like in vitro almost nothing works. In vivo, it's much more important to do it there. And then you pull out, you sequence what ends up in the tissue or in the cell, and then you can say, oh, you know what? Out of this 10 million variations that I tried, these 100 consistently ended up in the muscle, and they all have these motifs. So therefore, this must be a good way to target the muscle, for example.
Got it. Almost a form of directed evolution.
Yeah.
Okay, got it. And then obviously the other sort of less direct method is by manipulating the protein corona. But then what you're doing is you're engineering these proteins that then that actually serve as intermediaries that then recruit other plasma proteins that then themselves are selective for certain targets or certain tissues.
So that's also one way, but I think mostly what people do is, well, again, they screen. At least in AAVs, you can definitely do that. And you don't really need to know what you're doing if you can screen enough. That's one. But the other thing is, if you know that in my target tissue, I have a specific transmembrane protein. So it's not that it's in the plasma, but you just targeted that one protein. If it now binds there, there's a higher chance that it's going to be picked up by the cell. Right? So now we can do targeted rational engineering. Yeah.
Got it. Okay, fantastic. And I mean, obviously, your work has been relatively successful. This is a paper that hasn't been released yet, but talk about what you've been able to achieve in sort of whole organism gene delivery, and what sort of distinguishes this from past attempts.
So first I set out to try to hit the whole body to engineer longevity therapies. Of course, longevity aging affects every cell, so you got to hit every cell. And also, at least initially, I tried to hit like very equal biodistribution or expression across the whole body to sort of make it equal, right? And what I ended up discovering was no single AAV serotype or any vector for that matter that I tried was good enough in terms of giving you equal biodistribution across different tissues to a sufficient level. So what I ended up doing, long story short, is combining multiple different serotypes by picking them in a way that they would complement each other's strengths, if that makes sense. So, for example, AAV9 gets into the liver very well, also gets into the muscle and the heart pretty well. Some other places, it's fairly broad, doesn't get into the brain very well. Like, even though it gets better than some of the tissues, but the brain is still very, very low. There's a bunch of other serotypes that have now been engineered, like PHBB, PHBEB, that hit the brain at almost like 100% efficiency, right? And by that, I mean, not that everything goes into the brain, but rather at a high enough dose, you can get almost every cell of every brain cell, right? At least in mice. So by imagining that you have these different serotypes that have their preferences of tropisms, they all hit different tissues. Well, my thought was, well, if you just put them all together at the right ratio, can you make them work and can you make them into one unit system that's like a cocktail that you inject that hits all the cells in the body, right? And yes, that worked. But what I quickly realized was that you don't only want to do this at the level of the AAV serotype, you also want to have other levels of control to control what happens after the DNA enters the cell. And concretely there, the two other things that I ended up using was microRNA target sites. So with a microRNA target site, you can tissue-specifically lower expression in a given tissue, like the liver, for example, when it's oftentimes too high. And the other one is Kozak sequences. So Kozak sequences are the sequences to which the ribosome binds. And by altering their sequence identity, you can alter the strength of ribosome binding and therefore translation. So you can overall control how, like, what is the translation rate and therefore what is the expression level of your protein. And so the system that I ended up building in my PhD, in summary, was a combination. It's a mixed bag, it's a cocktail, putting these different elements together in a way that would work as a whole. And the key here is that when you have multiple different handles or components, now you no longer can custom it. Like, you can't just say, oh, I think dose of 1E11 per mouse is going to work. You actually have to know what dose and at what level you need to use. And that means that you have to map them out. And so that's what I had to do. I had to take all these individual components, test them in different doses and different tissues, map out mathematically how each one of them worked, and then try to see if I could model and predict, if I put them together in a certain way, that they're going to produce a useful outcome of what I want. And funny enough, no one had ever done this before. Like, in AAVs were discovered like in 1960s or so. In 60 years, no one had actually, to my knowledge, put mathematics on the expression curve. So we didn't even know if you could model this, like, was it going to be linear and so on. And if you use different versions together, were they going to compete? You know, was it possible to do this? And the answer there was, we're actually super lucky. AAVs behave very linearly. You literally just use a linear y equals ax plus b model to model this effectively. And at least the ones that I used, they didn't have a lot of interactions. So it's almost like 1 + 1 = 2, which we're very lucky with that. So yeah, they work super predictably, and therefore the modeling itself as an approach works.
Talk to me about these tissue distribution curves.
Sure.
What's on the x-axis? What's on the y-axis? Yeah, a little bit about that.
Yeah. So let's take, I don't know, like 5 tissues: brain, liver, skeletal muscle, heart, and lung. Right. Let's take 3 different serotypes: AAV9, PHBB, and I don't know, AAV5, or let's say AAV5, if I'm not mistaken about this. AAV9 gets very well into the heart, the liver, and the muscle. Almost not at all into the brain, and kind of poorly into the lung. Right? AAV5 goes very well into the lung, almost nothing goes anywhere else. PHBB, engineered for the brain, goes very well to the brain, and pretty poorly to everywhere else. Right? So now, if you want to hit equal amounts of DNA across all these tissues, if you select them in the right doses and put them together, these 3 serotypes, then your overall result is going to be equal biodistribution across these tissues. That's the big picture.
Got it. So you want uniform distribution. Each of them have a sort of like a triptych to different tissues, and then if you continuously map the dose response curves in different tissues, then you can combine them together linearly, which is the interesting point, to get uniform distribution in the end, right?
Yes.
And you want, like, most cells to have, more than 90% of the cells to have like 1 copy of the gene. You don't want multiple copies of the gene. Tell us about these other levers of control.
Actually, you do want multiple copies of the gene, or rather, if you're at 1 copy per gene on average, per cell on average, then you are facing, stochastic noise where some cells have it and some cells don't, right? So I'd say in general, it's probably a good idea to hit at least 10 copies per nucleus of your vector. And then you know for a fact that, purely by statistics, most cells truly have enough of your vector, of your DNA, to express this at a predefined kind of modeled level. Now, hitting equal biodistribution is one edge case of this system. Like, theoretically, you might want to hit different patterns of biodistribution. And actually, for most things, you do need to do that, because if you want to, for example, replicate the overexpression of a gene, and then rarely is it like equal expressed across all the tissues. So you might want to control that. But for longevity, equal distribution is, of course, the goal or very important because most cells are affected. Right? Yeah.
Got it. So you want to hit all cells relatively uniformly,
Yes.
especially if what you're introducing are genes to slow down aging because all cells age. And then, yeah, what are these other tools of control that allow you to manipulate gene expression and sort of make it even more uniform than just this linear combination of AAV serotypes?
Yeah, so I would say that you have 3 or maybe 4 major modes of control. There's probably more. There's a lot of things that happen in molecular biology that you could theoretically tap into. But the 3 ones that are oftentimes used is promoter, it's the first one. Then there are microRNA target sites, things that reduce expression, and then there's changing the translation efficiency through cosec sequences. Now, for the promoter, we actually decided not to mess with this because when you deliver something with an AAV, you already have a limited amount of sequence space. Like you either have 4 kilobases or you have 2.5 kilobases, 4.7 or 2.4 kilobases if you use a self-complementary vector. And self-complementary vectors are much more efficient than regular 4.7 kilobase pair vectors. So effectively, if you want a very efficient vector, you have 2.5 kilobase pairs only to work with. That's one.
And sorry, what is a self-complementary vector?
So self-complementary vectors is a hack of an AAV. So the AAV is naturally single-stranded, but as a result of this, as it enters the cell, it first has to go through the second strand synthesis, and then it also has to sort of what's it called? It has to either circularize or it has to stabilize in some way, right? And oftentimes this ends up being like end-to-end concatenamers that AAVs make these like huge tens of kilobase pairs long concatenamers. But the fact that it has to go through second strand synthesis in post-mitotic cells where DNA synthesis is oftentimes controlled is a major limitation. So whatever the theory, in practice what we know is if you use a certain smart genetic engineering hack to make the vector complementary, self-complementary to begin with, which makes it double-stranded DNA, as soon as it hits the cell, expression turns on, there's no question, and it just like goes right away. Like in a few days, you have like max level of expression.
Got it.
With regular vectors, it can take up to a month to get there, and it usually peaks out at a lower level. So.
Got it. So in post-mitotic cells, you just don't have the machinery being actively expressed to synthesize DNA. That the DNA polymerases aren't very active in these cells, and so if you deliver like a single-stranded DNA in these AAVs, they're not going to be turned into double-stranded form. The second strand synthesis isn't going to work, so it's not expressed.
Well, it works. It's just, again, empirically, it takes much longer.
Yeah.
And it doesn't hit as high of a level as in self-complementary vectors. Yeah.
Got it. Okay. So this like limits the size of the DNA that you can insert compared to the 4.7 kilobases.
Yes.
So now just like half the size, basically. But then you mentioned siRNA target sites, right? So this is you're adding in a sequence onto this DNA that is a target site for an siRNA that is differentially expressed in the tissues that you don't want your sort of synthetic DNA to be expressed, right?
It's not correct to say that you don't want it there. It's rather that you can dampen it down. And the number and the strength of these siRNA target sites will control to what extent it is dampened down. And of course, the type determines in what tissues it is being dampened down. But yes, in short, different tissues express different small interfering RNAs. And if you put a target site onto your vector that is complementary to them, then in the tissues in which they are being expressed, you lower the expression. Right? So we opted to do that instead because the promoter, the sizes can vary. Even though you can make them very tissue specific, oftentimes they can get fairly big, and also the strength varies. So I kept one short, very efficient promoter, used different siRNA target sites, and then also varied the cosec sequence to have these 3 levels of control.
Got it. Okay.
Yeah.
Interesting. These siRNAs, in this particular case, in this paper, you use them to dampen down expression in the liver. Is that right?
Yes. Yeah. Because that's where we ended up having more expression than other tissues because of the AAV vectors that I ended up choosing.
Okay. Yeah. So, landmark results, a great paper. I hope by the time this episode gets published, it will be released and that our audience can go and read it.
Yeah.
But what's left? Like, how do we go from this result to genome-wide delivery in humans? There's obviously like these like safety issues. There's this like kind of just general fear of viruses. I mean, there's 2 patients that died recently in an AAV clinical trial, probably related to the dosage of the AAVs. Yeah, like what are all the things that you're considering and you think need to be improved here?
Yeah. So first off, what could we achieve with this system? Well, across the body, on almost every single tissue that I look into, which is 24 tissues, we have above 1 copy per cell on average. And, it varies depending on dose, somewhere between 5 to 50 to 100 copies per cell on average. So now, by combining these things, we can actually hit the entire body. That's one. By looking at the protein expression, we can see that 80% of the body mass across like the entire body, again, across these 23 different tissues, express your target protein. And that includes almost 100% of the brain, 100% of the liver, 100% of the muscle, 100% of the heart, and somewhere between 50 to 100% of the remaining tissues, right? So the key thing here with this result is actually, for the very first time ever that I've seen, someone has showed that you can take an adult old mouse and truly put DNA across its entire body in a way that really works, right? That's like a proof of concept that we can actually do this. So that's one. Now, as a research tool in mice to study longevity, to study anything, it's super useful because, as I mentioned, if you can start out with an 18-month-old mouse that's already old, and you can make the vector, in which case, in our case, it takes about a month and a half to make it in a month and a half, you can have your entire study be done in like 6 to 7 months. Whereas before, you would have to wait 2.5 years for the mice to age and spend another 1.5 years to even make the mouse. So we're going from a 4-year timeline, in my PhD it was 7 years, down to 6 months or so. So it's a massive time saving, right? So as a research tool, it's super useful, and that's why I used it in my own therapies later on. What's left to do? There's a lot. Humans are different from animals, from mice in many different ways, and primarily our immune exposure. So many humans have pre-existing immunities to AAVs, and therefore they have antibodies. And what's known is if you deliver a high dose AAV into someone that has pre-existing antibodies, that can cause maybe a complement-based reaction, but basically like a very high runaway cytokine storm where even like a small amount of antibody that's there can cause like a strong immune reaction, right? And that's what's been happening. That's one thing. There's other things as well, like if you express a cargo protein that is somehow itself recognized by the immune system, like by these cells later on, they can target that cargo protein, but then as a side effect also kill healthy cells that are now expressing this protein. So you have to be careful when you with your payload and how you do this in the humans. But what are the 3 major challenges? Well, the first thing is the safety and redosability of things in humans. And there's actually a ton of progress there. So first, many people are engineering new AAV serotypes that haven't been seen by the human population before, and therefore we don't have pre-existing immunities. Right? So that's one. Secondly, there are now 3 or 4 different methods that can actually fairly effectively deal with the pre-existing like antibodies, or even if you get the first dose and you develop antibodies, then the deal with the antibodies that then are being produced against the AAV. Mm-hmm. And I can go into depth what they are, but basically you can either cut all the antibodies and generate a transient hole in the immune, like in your production to redose, or you can bind all antibodies and again generate a transient whole, or you can prevent the immune reaction from happening at all using these ImmTOR nanoparticles. So it's not perfect, but
These
there's
immunotolerant nanoparticles, you said?
so there is this thing called ImmTOR nanoparticle, where it's like rapamycin encapsulated in a nanoparticle that ends up in specific cell types in the liver. And when you do this, when you co-dose this with AAV, you can prevent, or not really prevent, at least based on the current data, but at least delay the development of anti-AAV antibodies that would prevent you from redosing it again, right?
Okay.
So otherwise, if you put in a large amount of AAV, you generate antibodies against this, then you can't redose again. So new serotypes that haven't been seen, preventing antibody response, and then removing existing antibodies if they're there to dose again. And of course, serotypes that are more effective for a given tissue to lower the overall dose. So this is all happening, that's one. So safety and redosing. Second part is manufacturing. So right now, AAVs are still pretty expensive to manufacture. There's a ton of work there. And like every year, the costs come down because better methods and so on. And I think at the limit, it can probably be as cheap as a biologic, although I'm happy to go into, I think, how this could happen, but that's the other part. And the third part is just better serotypes for humans. There's lots of serotypes for mice because mice are easy to screen, and sometimes this translates, sometimes it doesn't. And now there's very good work happening in humans. So yeah.
Got it. Like as an example, one of the serotypes in the study that you were involved with is very good at targeting brain in mice, but not so much in humans. Is that right?
That's right. Yes.
Got it.
Yeah.
So we just need just more serotypes, better engineering of these serotypes.
Yeah.
And then you mentioned enzymes that can be delivered to basically enzymatically cut all of the antibodies in the body to create like a hole for these AAVs to be delivered.
Yes.
And then these like other mechanisms to delay the generation of antibodies, allow redosing.
Yeah.
Then there's like very long-term methods that I've also heard is like, can we insert like in a safe harbor site, some kind of like a lock and key mechanism that allows us to avoid having to use AAVs to deliver genes in the future if there's like just like a more cell type specific sort of barcode on each cell. Do you have any sense on that? Or
I think it's possible, but I think there's enough locks and keys that we could take advantage of already. It's just it takes time and it takes people and a certain amount of capital. But I'm fairly confident that we can solve this. Now, this is like the basics of delivery. It's like, how can you get stuff into humans reproducibly and repeatedly in a way that's cheap enough and safe enough? But then there's the layer beyond that, which is if we can now do that and at the level in which I can do in a mouse, you can start building up large circuits, even though you can only deliver a small piece of DNA. And there, the question is just, how do we take something like a synthetic biology circuit that's normally like 10 to 20 kb, 30 kb or more, and break it apart in a way that it would function from these different bits and pieces as a one whole unit. And that is something that no one has ever done before because, well, guess what? The underlying tech hasn't gotten us there yet. But then that's another thing that's left. And related to that is the stability of things inside the cell. So what you mentioned, lock and key, and like, can we put a landing pad? So AAVs are episomal. They don't integrate into the nucleus. That means that if the cell divides, they get lost. To overcome this, you need to add an integrase, or you need to somehow get them into the genome. And that just means delivering another protein or co-delivering another protein. But if the first three problems are taken care of, we can hit all cells at high levels, and it's safe and it's cheap enough, then this is not going to be an issue because you can just add another vector that combines these and just run with that.
Got it. And like presumably we can stitch together DNAs once they are delivered. Like we do this in industry, we do sort of golden gate assembly and whatnot. Like is that feasible where we like deliver just like parts of the DNA that we want and then they get assembled later?
Yeah, it is. And that's what AAV already does now. Like it forms these head to tail concatenators because it has these ITR sequences, inverted repeat sequences that are used for packaging the vector into the AAV capsid, but they are very, like, it's the same repeated sequence. So in the cell, they end up recombining and making these almost like spacer type concatenators. And you can kind of do the same thing with your different vectors. I think actually that is probably less important in terms of what form they end up in the cell, so long as they integrate and they're stable, if that's what you want. But rather, how can you use these 2 to 4 kb size bits to reproducibly and accurately and predictably engineer a whole circuit? You know, and again, they don't need to make up one big giant, but it's just you figure out how to break it apart.
Got it. Yes, because let's say you have these enhanced genes that you think will improve lifespan or just like reduce cellular entropy and aging. You want the expression of these genes to be regulated in a precise way, similarly to how every other gene that is currently integrated in the genome is regulated, right? So this is where gene circuits come in. You want to have like a sensor, a control side, an integrator, to basically manage the expression of these and also to perform computation, right?
Exactly.
Like
Yes.
similarly to, our we have high blood sugar, we need to release insulin, or we have low blood sugar, we need to reduce insulin, and various other like genes that we modulate. Is that a good summary?
Exactly. Yeah.
Okay.
I mean, at the end of the day, in order to engineer new capacity, which is what we need to do to push back aging, is really just, we age because the systems that we would require to stay young for longer just haven't been built by evolution. They just don't exist because, we live 3 times longer anyway than we used to live in the wild. Right? You have to have the capacity to just do proper, genetic engineering with sensing and circuitry and everything, and there's just no way around that. Yeah.
Got it. Okay. I think we'll touch on the manufacturing part another time.
Sure.
But I want to move on to the rapid fire section. So I'm just going to ask you some questions and tell me what's the first thing that comes to your mind. The first one is, if you were the president of the US for one day, what administrative change would you institute in order to address longevity bottlenecks?
For one day, man, I don't know if you can even get a bill signed in one day. But okay, if I was a president for one day, I would probably use a discretionary budget to do a massive ad campaign to get. So that's one thing, but also just push a lot of funding into the longevity space, so that people actually can work on that. But massive ad campaign to get all the bright minds and like all the high school kids that are trying to figure out what to do with their lives into longevity, because I think that's where you can probably make the biggest bang for the buck in terms of effect on someone's life.
Got it.
And I would argue that a big issue that we have is still like the limitation of brain and talent in a space. Arguably, that's the limitation anywhere. And yeah, if we can get more people in and have them funded, then we're good.
Got it. Like the Manhattan Project or the Apollo space program, but for longevity.
Yes, maybe more like Taylor Swift for longevity or something like that.
Okay.
Get people excited en masse.
Understood. Okay. So the other thing is you spend a lot of time, I think, traveling back and forth between Boston and San Francisco. You're based in the Bay Area. Why? And what's your sort of favorite quality about the different cities?
Yeah, I think both of them are excellent. Also, both of them have their downsides. I ended up moving from Boston, so I did my PhD at Harvard, to Bay Area because I thought that with Oden, which is where we automate animal studies, originally the target customer was the longevity biotechs that were doing longevity studies, and many of them are there. So I thought, look, they're there. I gotta be next to my customers, so let's move there.
Got it.
But yeah, Bay Area is very strong in big picture thinking, crazy ideas. Boston is very strong in executing, like low on the ground and like very scientifically on bio particularly. And I think put them together, you get a good thing.
Got it. On that note, Altos Labs, Retro Biosciences, or New Limit? What's your favorite company?
I'm not gonna pick. They're all good in their own ways. They're trying to do different things. New Limit has done a ton of very impressive progress on just technically screening these things. Retro has probably recruited the highest concentration of talent that I've seen anywhere in the longevity space. And Altos has, I suppose, brought in a lot of people from standard academia and funded them to do what seems to me to be fairly basic fundamental longevity science. And I mean, of course, Altus's approach is the longest running of these, like, you need a long time horizon for it to pay off. But I think they all have their strengths. So, yeah.
Okay. What's one book that you'd recommend to our audience and why?
I would say actually Nikola Tesla's autobiography.
Okay. Oh, I didn't know he wrote an autobiography. That's interesting.
He wrote an autobiography. Reason why is because if you read his way of thinking, like, he was very advanced for his age. Like, he was ahead of his time by arguably like 100 years or so. But he developed engineering to an extent that it seemed like magic to his contemporaries. And I think that should be the mindset, which is like, how can I make such an impact? And how can I really push the envelope so far that for other people, it seems like this should not be possible, but it is. And I think that's the right frame of mind to think about problems and then kind of bring it down to earth and figure out how to actually execute on that. But yeah. Got
it. Didn't he marry a pigeon, or was that Isaac Newton?
No, he didn't marry a pigeon. He was afraid of women. So pigeons were a companion of his at the late of his life.
Okay.
But yeah.
Awesome, Michael. It was great having you on the show. Thanks for coming on.
Yeah, likewise, man. Appreciate it.
Okay.